Computer models and simulations are important tools to build medical devices and therapies. In order to build reliable models, we need to have input parameters, such as tissue material properties. Currently, there is no consensus on how to measure these tissue material properties. The Virtual Physiological Human Institute, together with the Avicenna Alliance and Fiber, are taking the first steps to reach this community consensus and to build a library of reliable tissue properties.
There are several types of physical tissue properties, and we'll start by tackling mechanical tissue properties. And instead of dealing with all aspects of tissue characterization at once, we will focus on a single aspect per test campaign. And we start with uniaxial tensile testing. In phase 1, we'll quantify the methodological variability that's currently present between research groups. That's why our team has prepared 25 identical sets of porcine aorta under carefully controlled conditions. The packages are shipped per express delivery in dry ice to reach 25 expert labs from all over the globe. These labs will test the same type of material using the same type of test, but with their preferred methodology.
In the second phase, we will collect these results and determine a consensus methodology. Afterwards, the participants will be sent another sample set and asked to repeat the measurements using the consensus methodology. This is phase 3. Bringing together these results will allow us to determine whether the use of the consensus methodology has led to a reduction in the methodological variability. In the final phase, we will share the results, the tissue property library, and the consensus methodology with the entire community.
Check our website c4bio.eu to follow the progress of this exciting initiative.